Let's begin with the question whether there are questions from the last lecture pertaining to the topic that was brought in the lecture. The slides should be online today. We have somebody who has volunteered to do the videos. I haven't said hello to her, but the video is running. I assume that the video of the lecture today will be online within the next week. And thank you. Is she here? She's hiding. Thank you. Okay, let's do a recap. So coming to life is DNA, are proteins, are cells. Cells are the larger entities that are coming to life. And coming to life is the process, the information flow that flows from DNA being the information, the storage, the library, to RNA being an intermediate product essentially, to the proteins that are the machinery of life. And DNA and RNA is a 4-letter alphabet. Protein is a 20-letter alphabet. In order to get 20 different amino acids with 4 different nucleotides, you need to use triplets. That, in fact, is this codon wheel. So the first nucleotide, 1 of 4, is here, the second and the third. And the example I picked last time was AAA leads to the amino acid K. And you see that some of these amino acids here are coded by more than one codon, so more than one triplet. Most of them are coded by more than one. Methionine here is an exception. And then there are 2 additional things here that are not amino acids, stop codons. That essentially means stop reading. And that is essentially the end of the gene. That's the end of the protein sequence. You can determine these sequences for any organism that you may encounter. This is a simple sequencing machine that is roughly, you can imagine it like a printer, and that sequences essentially one person per day. Now, there are about 20,000 proteins in human. So again, the machinery of life are proteins. Every cell has the same DNA, but different cell types have a different makeup of the types of proteins they use. So I said there are 20,000 in human. This means in all cells together we have 20,000 proteins. In most cells active are 10 to 14,000. So this is a subset of these 20. Not all 20 are used in all cell types. And that makes cells different. So the way they use proteins. Overall, and the number is wrong here, by an order of magnitude, currently we have 111 million protein sequences known. So this is not 11 million here, it's 111 million. I missed one. And the distribution is such that the shortest protein is about 35 residues, the longest 35,000 or 30,000. And in between is a lot of variety. Just a sec, let me finish my sentence. Proteins are typically, domains are typically about 100 residues long. Yes, question?
A protein and a protein sequence is the same thing?
So the question is whether a protein and a protein sequence is the same thing. Yes, so the protein is made up of a sequence, so amino acid sequence that defines this particular protein. Then it's put into a solvent, it adopts a unique 3-dimensional structure. Now, which of these 2 things do you call the protein? Do you call the sequence the protein? Do you call the structure the protein? The actual functional thing acts as a thing in 3D. But it begins its life as a sequence, and essentially so Thursday is skipped, May Day is skipped, but next Thursday we will begin to, in fact, compare sequences. Proteins come at different sizes, and these different sizes here is just differences of these sizes where the scale on this one here is even reduced. The cell has an outside and an inside. Around the cell is a membrane. That membrane essentially distinguishes between inside and outside. And in some sense, if you want, that's sort of the point. The membrane is the point where life defines self. Everything inside it is that cell. Everything outside is sort of the environment, is not self. And today, I'm going to give you a little bit more about the energy of amino acids, the way proteins bind and come together, about domains and 3D categories. But let's begin, because a lot of what I do is about images. And I show you images of proteins, and very much like this question, is a protein sequence a protein, or is a protein structure a protein? The images that I put here, they give you this idea that what I show is a protein. It's an image. And I'm trying to go through a couple of images to make this point that it is all an image. So we often believe that we understand, and we sort of represent truth in images, but they are not the truth, okay? So the slides come from Marco Punter, who is now in the Cancer Institute in London, and here's the first one. Does anybody have any idea what that is? This image, so yes, that is a person, that is not what I mean. This one here. Anybody? Any guess is fine, come on, this is art, yes?
Houses behind trees. There's a tree and there's a house.
Houses behind trees is what he says. And just Georges Braque, houses at the Lastac, houses behind trees is exactly what it is. So you could have guessed the painter, you could have guessed on top maybe the area or the region where that is in the south of France. But essentially that's exactly what it is. Houses behind trees. And I have to say that you sort of spoiled the game a little bit here. Nobody ever was so right on. Actually, I was looking for the name. Let's try. Now, so there are images, and these images, I don't know what it did to you. Maybe some of you had this idea of a house, maybe some trees, whatever you had. This is not a photograph. This is not a photographic image, but it portrays in your head a mapping of something that you have seen that may have been exactly what the artist saw. So, and the very same thing here, this in fact is a cell. I said that already briefly. The green thing here signifies the lipid bilayer, the membrane that closes the cell or distinguishes the cell from the environment. And the inside, this happens to be Mycoplasma genitalium. So one of the smallest organisms with about 500 proteins or 500 genes. So one of the smallest organisms that exist. Now, what this image here from David Gutzel here is showing, and it's a drawing. And it's a drawing because this we cannot see on a microscope. All right, we can stain parts of the cell, and then we see some red regions, green regions, and we see part of it. We cannot see it this way. The way David Gutzel is doing it is he reads a lot of literature, and his profession is to, in fact, make science such that you can imagine it in a way which is closer to the truth than the photographs that we have, which essentially would be photographs of something that, for instance, is only the nucleus stained. So there's going to be some blue in the nucleus. That's a photograph of a cell. But what this one shows is all different kinds of elements. Now, the important part of this image here is if you imagine a cell to be a plastic bag filled with water, and then there's some material swimming in there, that is clashing with this image. How so? Why is what you see here not quite fitting to this idea that you have a bag, a plastic bag filled with water, and then proteins swim around it? What is clashing in what you see here? Why does that not fit to what you see? Can anybody? It's trivial. Can somebody say it? Yeah.
It's an image that's static.
See, the image is static. Yes. Oh, that's a very, very important point. I take your point. So the image of a water bag. The water is constantly flowing. I cannot see that because any photograph will not show this flooding water. That's true. But there's something else here that is clashing.
And there are no pores.
There are no pores. In the plastic bag, they are not pores. Oh yeah, fair enough. So she says the plastic bag is tight. And this is very, very important. The lipid bilayer lets energy in and out. So there's constant exchange with the environment. You could not build life upon on a plastic bag. So let's get rid of the plastic bag. But this is a different story. That is true. But I mean something else on this image here.
It is very dense.
It is?
The image is very, very dense.
That's the point that I mean. He says the image is very dense. Yes, what you see here is that a cell is packed with stuff, whatever that is. before I name it, and that in fact is true. So this drawing of David Goodsell here is really realizing or showing a reality. You can imagine a cell more like a watch inside than like a water bag. Life works only because there's water in there, and the proteins fold because they are surrounded by water. But it's still extremely densely packed. Okay, so there's just a little bit of a layer of water. It's not this idea that you have a bag of water and there's a lot of freedom to move around. It's very densely packed. And that is true for a cell. This is an image of a eukaryotic cell, or a cut through a eukaryotic cell, which is essentially like a prokaryotic cell, like a mycoplasma, just bigger with subcompartments. What is this? So it is, before anybody says that, is red and blue. Yes. Anybody has any idea what that is? The only way you can have an idea is that you're already ahead, that you have seen this sort of thing before. There's no association, so this is not houses in Munich or something like that. So this, in fact, is a protein, where now every ball in here is one amino acid, or is one placeholder for an amino acid. So an amino acid consists of different atoms, and if you sort of take the main center of an amino acid and put a sphere around it, then you get this type of image. Okay? Now, you see a lot of balls. Okay, so there are a lot of amino acids, and maybe it is difficult to sort of see regularities. You see a red and a blue, but you don't see anything about the relation between the two. Now, there is a repetitive element in 3D structures called secondary structure. It's repetitive in the sense that when you are used to seeing these images here, you sort of can make out that there are two repetitive elements that you see again and again and again. One of those is called a helix. A helix essentially goes like this, right? And in the next image, I'm going to show you the same image here, except for I'm going to highlight the helix, and I'm going to put or show a rod where the helix is. So the rod is not reality. It's just highlighting here is a helix. So it's tracing these balls here and sort of putting a rod around the balls that form a helix. That's this image. This is not more reality. In fact, you could argue this is less reality than on the image before. But it immediately shows you something about symmetry of this molecule. You immediately see that there is something, even if you haven't seen a protein before, you start to suspect that there is some symmetry between blue and red. That blue and red actually are similar repetitions of something similar, all right? You couldn't see that before. Or you would have to, anybody in this room who has never seen a protein, very few people in the room who have never seen a protein would have seen that on the image before. On this image here, you see it immediately. Okay? And that's only a visual help. So this visual help that is not reality helps you to see something that is reality. And that brings up the question, of course, which one of those is more true? Right? None of them is true. They reflect different aspects. They show different things. When you look at larger proteins, this is the so-called alcohol dehydrogenase, ADH, and this is a molecule that is important for those of you who like to drink beer and feel good at the next morning. There are some people here in the room who will actually lack this enzyme. And because when you lack this enzyme, then the alcohol consumed is much more dangerous for you because you essentially cannot recover easily. ADH heats up the alcohol. ADH5 is in fact, again, coming together. You may begin to sort of appreciate, and by the way, the helices are now shown here green or orange as these sort of helices. And then you have these arrows here. That's the secondary structure element, beta strands. If I gave you more time and we could play around with this on the screen, you may be able to see that in fact what you see here, ADH5, is made up of two subunits that are again repetitions of the same thing, like in this molecule that I showed you before. That, by the way, was a different protein. And in fact, it's a dynamic object. So that brings us to the next image here. What is that? And I can tell you, this time it's not a house, it's not trees, and it's very unlikely that anybody in this room who has not seen that image before guesses what it is. Yeah?
Paper burning.
Paper burning. So paper burning is in fact one important element of the answer of what this is. Any other association? So what I'm going for here is really associations. Anybody?
Light reflecting on a glass.
Light reflecting on what?
On glass.
On glass. So light reflecting on glass. Maybe you see those here. Fire, light reflecting. And both of those capture something relevant. Anything else? Yeah?
Clothes soaked in dirt.
I'm sorry, I didn't get it.
Clothes soaked in dirt.
A cloak soaking duck? I'm sorry.
Clothes soaked in dirt. Cloth soaked in dirt.
Oh, that's interesting. So he's talking about me jumping into some pool of dirt and then being very dirty. That's an interesting one. And actually, there's some truth in that one. Okay, so Umberto Boccioni, dynamism of a soccer player. And I understand that not everybody in the room 100% got this right. This is an artist rendering of that photograph. It's just the same thing. It's just the artist. My point here is, so somebody said before that since I have a frozen image, I cannot see the movement. Okay? So some of you may see soccer or may watch soccer, and maybe you're sort of familiar with this player who no longer plays, but maybe you sort of see in this image that this guy is very mobile and that he's very fast and very agile. But if you really don't know anything about soccer and this player, then essentially you see a 2D image, and the 2D image is a frozen moment in time, and there's no dynamism in this image at all. You don't see movement in this picture, really. I mean, there's a ball, and somebody's going to kick the ball, and there are some other people who are in the way. But this rendering that some of you may say is not a one-to-one rendering of this photograph gets in your heads issues about fire, energy, about light reflection. Again, this is something that is very mobile, or movement. About the guy falling into the dirt. I'm sort of mistaking your cloth soaked in dirt. What I'm trying to say here is this image transports some of the ideas of seeing this guy play. Okay? And again, it captures a different reality, and it captures in some sense a different aspect of reality, and in the very same sense. So here we have two different realities captured in two different images. You can argue that this one here that goes where every line here is representing one particular three-dimensional structure, and this is superposition, so an overlay of many of these three-dimensional structures. So you cannot really easily count how many, but 1, 2, 3, 4, 5. So you see there are many. And this one here sort of is this summary, and the summary shows you the arrows are secondary structure segments. And both of those capture different realities. None of them really is true. Both of them capture different aspects of truth. And I'm now going to go into issues about energy. It's the same thing. In physics, you learn that the energy is relevant for certain things that happen. When you talk about the way Earth attracts Moon, then the energy is so simple that this simple image actually is right. When we talk about the universe of proteins, the story gets so complex that the image is no longer right. It's an approximation. And another image captures another aspect of that truth. So let's begin with the constituents, with the amino acids. So proteins are made up of amino acids. Those are the parts. This is one particular amino acid. An amino acid has sort of two major parts. What is called a backbone. So it's main thing, and this main thing is the same for all 20 amino acids. And then in red here, it has this side chain. And that is the part where the different amino acids differ. Okay? As simple as that. So it has something that is the same for all 20 amino acids, and then something in which they differ. Now, when I showed you this ball on the first image of a protein structure, essentially that is a ball of this, what is often called the C alpha, the main carbon in a protein. When you join two amino acids in something that is called dipeptide, or in a peptide bond, so dipeptide is two amino acids, you join them, one end, the CO binds the HN on the other end. And you see that the side chains, the way this peptide bond forms, the side chains are on opposite ends. Right? So the first one has, in this particular image here, the side chain up, and the second one has the side chain down in this image. And the bond is favorable, it releases energy. So there are two forces here. One force essentially is sort of pulling, and one is sort of the electrostatic force is pushing away. But overall, this is energetically favorable, provided you get close enough. So you have to sort of push a little bit, and then closer you cannot get because there's another energy barrier. Now, when you look at the way these bonds form, one way of sort of reflecting this bond is by saying, okay, we have these C alphas here, this C alpha and this C alpha, and they are somehow bonded. Okay? And this is the way you can sort of look at a bond. This is another way you can look at a bond. And this is yet another way where it may not be visible for all of you, but it's essentially the electron density shell. So where at some probability the electrons are moving around. These are three different images of the bond that forms. Okay? Again, none of them is really right. Bless you. And then dipeptide is just two amino acids, and then you simply keep going and build up amino acids. And you join these into a protein, just like that. Now, when you join amino acids into proteins, you can rationalize some of the things you observe by simple features that you assign to amino acids. And again, these simple features are features that in chemistry you would learn, this amino acid has that feature. But when they are coming together in a protein, these features are not individually measurable. One way of saying that is, one of these features I will show you is charge. If the feature would be cast in stone, then any book you find will show that the particular amino acid has the same charge, and that is not the case. So there are different ways to approximate what the charge actually would be, or the hydrophobicity, or other features. So let's just dive in. So those are the 20 amino acids here, 20 different here, and there are two of these, the R and the K, that are positively charged. There are two that are negatively charged, the D and the E. There are 5 polar, so polar means so there is some sort of pulling apart of the energy cloud. So part of the energy cloud is more on that side, part of the energy cloud is more on that side. Again, and what I don't show is the hydrophobic ones. Hydrophobic one, water phobia is, as you may know, being enemy of, phily is friend. Hydrophobic means you want to stay away from water. Some amino acids are hydrophobic, so they cling together and stay away from water. The protein folds, so adopts a unique three-dimensional structure that is called folding. Folds in an aqueous environment, so in water. Okay? So hydrophobic amino acids then will be inside of the protein, will stick together. Hydrophilic amino acids will try to be happily close to the water molecules on the outside. An essential domain, or an essential part of a protein structure is a domain, so units. And again, I said one way of thinking about those domains is that they fold independently. So you would cut them out and they would form their own protein. There is, I'm going to say this now, but for the rest of the lecture, this will not really matter anymore. So there is a DNA, there's some detail on the level of the DNA. So DNA has exons and introns, red and blue. And the introns are regions on the DNA that are actually cut out. Okay? So only the red ones are joined together to really form the proteins. Meaning that there's a lot that we carry on our genome that's not actually used to build proteins. This a lot is really a lot. So about only 1% of our genome is this red part. That does not mean that 99% are the blue, but in fact there's another part that is in between genes that makes up most of it. The blue ones in our genome make up as much as the red, roughly. So it's another percentage, and 98% are somewhere in between genes. And for many years that has been coined the junk, so something that is not needed for life. Clearly we see signs of other proteins in there that are currently not used. We see signs of viruses in there. We see a lot in there, but we also by now believe that there's a lot of signal in there that tells the machinery, the proteins, how to translate the information. How exactly that works, we don't know yet. How much of this 98% that used to be junk actually really is junk or buffer, we don't know. But what clearly we believe now is it's not junk. Or there's a lot of information in there. Now, what we see when these exons, the red ones here, come together, in this particular case, you see there are 3 of these reds coming together. I said proteins have domains. So when people discovered this fact that exons have to come together and that they're protein domains, the first idea, the first idea was maybe the red ones relate to domains. So maybe a continuous region, a continuous red region of the DNA will form one particular protein domain, and that is not true. Yes?
So exons and introns are also sequences of amino acids?
No. So I'm sorry. So the question is, exons and introns are sequences of amino acids? No. The gene product here is the protein. That is amino acid. This here is on the level of RNA or level of DNA. So this is nucleotides. This is before the splicing, before we are translated into amino acids. So the moment the protein is translated into amino acids, there is no splicing of this type going on. It is true that when I form a protein sequence like this, sometimes some region here is cut out. And this beginning region, given the chemistry, the first atom is an N, the last atom is a C in a protein chain. So proteins are labeled N to C, so begin to end, the begin is the N. So that's the beginning of the protein. So the N terminus here, the first part of the protein is cut away, and often, or in some cases, this first part is relevant to bring a protein or to regulate transport. To make sure that this particular protein, for instance, goes outside of the cell, goes through the membrane. And there sometimes is a stretch that can be fairly extended, can be 70 amino acids long, that will essentially only serve to go outside of the cell, and then it will be cut. And the real protein that lives on after that is the one from here to here. In that sense, there's something like splicing going on, but it's something very different. Okay? So again, up here is the nucleotide sequence, here's the protein sequence. So when proteins merge, one way we see this is we have a particular prokaryote P and a protein A and another same prokaryote protein B, and then you have a different organism, P2 here, where you have a protein C that merges these two. So where you have a single protein, a single gene that is twice as long, and that are these two together. And in fact, when you see these things, that's exactly the moment when you believe, if I have this one and I find two separate proteins that essentially align to look like it, then maybe my protein C here has two domains. Because in evolution, it just came together. Can you guess why why would that ever happen in evolution? Why would they not? The assumption typically is they begin like this. So that the assumption then would be that organism P here is earlier in evolution than P2. So why would possibly an organism that comes later join two proteins? Any idea? So to optimize, I'm not entirely sure whether that's what you mean, but maybe to optimize the way it folds. Yes, that is a suspicion. But remember, the way I think about domains is that in this particular case here, the 1 and 2, in this particular case, these two domains would be such that if you take them apart, they would fold alone. So the relative proportion of hydrophobic and everything is such that both of them are energetically somehow happy. Well, it could be that they are not quite happy. This could be an argument, right? That they're not quite happy and they are happier this way. True, could be. That is rarely the case. Any other argument? So, what happens very often is that in fact protein A and B have to somehow act together. This could be because they have to bind, and this could be because something binds to A, something binds to B, and that has to happen local, meaning that the binding events have to be somehow coordinated. So for instance, you could imagine that something binds to A and something binds to B, and these binding partners do something together, so that's why they cannot be that far apart. If you put A and B into one particular protein, they're already together. In this organism here, they first have to meet. Okay, so you add one level of complexity, and by joining them in one particular point in the genome, what you achieve is that you save time, or you reduce the complexity of the problem, right? And this, in fact, is what we observe very often. So when you look at 3D molecules, and these are two different proteins here, you may see that there is a region. So again, these are the helices, the yellow ones are the beta strands, and you may somehow guess that this part is repeated in the bigger part. So you see repetitions, and this is somehow like the one before, but in this particular case it's not only two, it's this one here has more than the 2 units that I showed you before. And in fact, when you sort of put them apart into different types of units where every of these symbols here indicates one domain. So some proteins you immediately see here have many domains. When you, again in the light of what I showed you before, this is in some sense you can find domains. So what I'm showing you here is protein A, B, and C, and D, and so forth. And the way I show them, the fact that A runs from here to here and B runs from here to here means that when I look at the sequences between A and B, I feel that this region, the sequences are similar. Could just be the identical word, right? So you have a region where B and A look alike. And then you have one, in fact, where C, B and A look alike, and so forth, and so on. And if you do this, then in fact you may guess that where you have many that look alike, they begin with this region, they end with this region, they look alike in that region, and since many different organisms have that, or many different proteins have that, maybe that in fact is a domain. And this is a way in which you sort of can guess domains from sequences. In this particular case here, this one would look like that, the other one may look like that. When I show you overall what fraction of proteins have single domains, then the answer is the blue one is the truth here. This is a different database, let's just stay with the blue here. Less than 30% of the proteins have single domains, meaning that most proteins have more than just one single domain. Okay, that brings us to the next issue. The next issue is how can we compare three-dimensional objects? The principal idea is somehow we have to match shapes, as simple as that, right? So we have to have a machine figure out that this one here fits into one of those 4 holes, and that in fact this may not be the best solution, unless you have a hammer. So how do you match? The first thing, so essentially the question is, does this red one fit here or here? And the first thing is you sort of find the best superposition, the best point of turning this here or there, and then you find a bunch of points that are corresponding. So you put yourself to a single point here, and then you ask this point and that point, and this point and that point, how much do they differ, and you sum over the difference, right? In this particular case here, we have a tiny difference for 2 points. And here too, I'm not entirely sure what I tried to say with this many images, but again, you have to label them, and then once you have labeled them, these points here in these 2 units, you can simply sum a root mean square deviation. So you have, in this particular case, in my image here, I have 8 different points, corresponding points. In the protein, these 8 different points would be the 8 amino acids. And for every single amino acid between 2 proteins, you find the corresponding points. Finding the corresponding points means that you superpose them, and then you simply ask, what's the difference between all of these amino acids? You square it, and you do a square root of it to get sign independent, right? So in this particular case here, you would have 2 different solutions. And again, first you have to find the point, and then superpose. Now, in this particular case here, the problem is a different one. In this particular case, they may fit, but you would have to scale. Scale you cannot do. You cannot blow up a protein. Amino acid corresponds to an amino acid. But it still could be that there is a theme where you have 8 different amino acids, and you find those, but then you have to cut them out. For simple shapes, scaling is easy. For proteins, this is more complicated. But this brings, yes?
Why do we have to do a 3D comparison if the proteins are just sequences?
So the question is, why do we want to compare 3D shapes and not sequences? Today I talk about the comparison of 3D shapes, and that will, over the course of this lecture, be the only time that I talk about it. Most of the time we talk about the comparison of sequences. This is not because comparison of sequences is good or better or easier. It actually is easier, but if I could do this here, if I had my 3-dimensional shape, then that is better than the sequences alone. Why is it better? Because proteins act as 3-dimensional molecules, and if I see how they look, I can guess at what they do and how they do it. If I can compare the 3-dimensional objects, I learn something about their function. I learned something about the dynamics of the molecule that is not visible from sequence alone. But for most proteins, we do not have the 3-dimensional shapes, so we cannot do this. Nevertheless, it's an important technology, and it's a very sort of intuitive one. There are all kinds of glitches, there are all kinds of problems. But in principle, it's very, very, very simple. And what I showed you here before is essentially one simple way in which you already have it. We get now into the next problem. And the next problem really is the one between local and global. So if you wanted to compare these 2 features or these 2 objects here, one way would be that here are 2 different ways in which you can optimally superpose them, right? This is one way, this is another way, and I'm not saying that I found the only ways. There are many, many other ways, most likely. But you also see that both of those do not fit very well. And you all immediately, intuitively see a way in which they would fit much better. All I would have allowed you to do is to cut. If you take scissors, so if these were paper objects, right, paper mache or something like that, you could simply cut them apart, and by cutting them apart, you immediately see that you can make red and green fit to 100%. These essentially are built up the same way. I just connected the green and the red are just connected differently, but they are made up of exactly identical units. In the universe of proteins, the domains are the same between the 2. It's just the way they are connected that is different. If I want to find a global superposition, I fail to have a good one. If I take a local essentially means allow me to cut, I find a very good superposition. But now I cut into units, and the problem with cutting into units is, well, I can just continue cutting. So I said that I find that these two objects, the red and the green here, are similar only once I'm allowed to cut. When should I stop? So the way I drew these images here, you immediately sort of recognize there are triangles, there are circles, so they are sort of larger units, and that suggests to you a way to cut. To keep these large objects, right? But in principle, in the universe of proteins, you could call them domains, but then you see that between domains you see subunits that actually are similar. And then you begin asking, well, why not cut into a domain? Why not compare three-dimensional objects on a level smaller than a domain? For instance, on the level of a helix. I showed you there are these rods, these helices that are found in many different proteins. So why not just cut out rods, helices, and compare protein structures based on these helices? Okay? Or put it differently, how to decide where to stop cutting? From informatics, you may find that the answer to this question would be, go out there, hunt for your expert, and the expert will tell you where to cut. Not the case. Can you think about a way to find a definition of where would you stop to cut? How could you do this? So there's no expert who's going to give you an answer. So the question ultimately is, you have a particular way of cut, and I'm asking you to answer to me, how would you test whether that cut, that unit, that smallest unit that you want to use for comparison is relevant or not? How could you test? Anybody? It's not simple.
Find the largest chunk of similarity and stop there.
So the largest chunk of similarity, so maybe not everybody heard it, find the largest chunk of similarity and stop there. Let's get back to these two guys here. Maybe the largest chunk of similarity would be one of these regions here, and you see that green and red you can align if you stop essentially as these triangles wait a minute, I had this somewhere here. So the triangles, the square, and the circles. And stop there, because between those two, you find a fit, right? And that is one way of doing it. Now the problem, that in this particular example here, the algorithm is completely tight. That's it. That's the right way of doing it. The problem is, what if I gave you an object that is different from this? So an object that does not have triangles, that does not have circles, and a square. In that particular case, your algorithm would keep cutting, okay? And would keep cutting, and then we would get to the question, is this cutting right? Or are you cutting into a region that no longer makes sense, yes?
Do you know the final structure?
So you, again, be careful, be mindful. You do not know what the right solution is. So for the two proteins that you're comparing, you do not know whether they are really fitting, because you're supposed to develop a method. There's no expert who tells you A and B are similar. Your algorithm is supposed to find the cutting and find the way of saying, this in fact is similar, this is not. This is too big, this is too small. And too big, too small in terms of cutting. How could you do that? No, we don't know what the end is. So we don't know what the right answer is. Is that what you asked?
And no. And when we did, we could do every cutting that is possible. And then do we know at the end that we have the right solution?
So the question is, if we really do all the possible cutting, do I then know what the right solution is? No. That's just the trick. I want from you the trick that gives me the answer, what is the right solution? Distinguish between the right and wrong. Nobody else can give it to you.
And define a new measure of similarity, which not only takes into account how much they overlap, but also kind of penalizes how small the cuts are.
So we're getting into an optimization criterion that makes it. So she says when we look at the similarity between two pieces, we also penalize when they are too short. So if I take an individual amino acid, then that is too short, and that has to be penalized somehow, right? How could I find the way? So I sort of find a balance between the degree of fit and the degree of penalty for cutting too much. How could I sort of adjust this balance? Any idea? Essentially what we need is a data set of things that somehow fit and things that somehow don't fit. While in every single pair you do not know the answer how far you have to cut, you can hope that there is some way of defining the valid unit here through scientific significant or statistical significant. So we somehow find units, we find cuts, we find the penalization, we find a score that allows me, so when I sort of do everything against everything, I get a distribution that looks like this. And when I look at this final score that sort of merges penalization and fit, then I'm far away from the blue, from the background distribution. So plotting something like that does not necessarily mean that for every A and B I know it. It only means that I know there's a bunch of background. There's the blue background. So most things somehow don't fit. This is what I essentially need here. Yeah?
I have a question. Do the units represent individual amino acids, or?
So I'm not entirely sure that I understand the question.
So you split into units?
Yes.
And these units represent amino acids?
No, so these units that I'm talking about must be something much bigger than an amino acid. Because when I'm sort of comparing on a level of amino acids, the story is trivial. Then I'm essentially back to single sequences. So the moment I come back to the colors, to amino acid, I'm throwing all the information from 3D away. So this can, whenever my method goes to the level of amino acids, I know I cut too much. So that's the point where I have to penalize somehow. What we do know it's smaller than domains. So in my people that I showed there in the green and the red, the triangles, those were meant to be domains. So we have to cut more than domains. But how far we really cut depends on details and depends essentially on the background distribution for that particular molecule, for that particular cut. So it will be something much larger than amino acids and smaller than domains. Okay? But this is essentially an optimization criterion that you will have to do. Now, one way of doing this is in the RMSD way, so you optimize an RMSD difference, and one way in which you sort of can compare the distances between two objects, in this particular case here, of 8 amino acids, is by looking at two 2D maps, distance maps. So here are the 2D maps, and essentially I can compare these two objects based on the 2D map of these two objects. Is that clear to everyone? So each one of these distance maps here simply takes the 8 points and plots for every single point how close is that point to all the other 7. Okay? This and that object. And then I'm going to compare two matrices instead of two 3D objects. Okay? And that is simpler because on the level of a matrix, I immediately know how to compare elements in the matrix. Okay? And I can also break it down much easier. Essentially, you look at sub-matrices. So this is Patrice Köhl, UC Davis, and the following slides are from him. Again, I'm repeating the same thing in order to do a structural alignment here between the blue and red and the green. So we either align the blue to the green or we align the red to the green. And you see that the first step really is to find the best superposition. Once you have the best superposition, you can compute deviation, the RMSD. And in this particular case here, you have two different deviations. For the red, it is, for the blue, it's 2.8 angstrom. So an angstrom is 10 by the minus 10 meters, so it's a nanometer here, 0.23 roughly 0.3 nanometers. this one here is a similar distance. The blue is as similar to that roughly as this one here. So the difference is similar. And that shows you somehow they fit alike. What is not considered here is somehow the penalty issue or the number of residues that fit. This is an overall shape difference. The blue has a different length than the red, and you should take that into account. Anyway, so these are two different fits. The one that I showed you before is a method that sort of compares 2D matrices. And the 2D matrix comparison brings me back to sort of a more generic way of looking at 1D, 2D, 3D. So when you look at a protein, you can look at it in 1D, you have a string of sequence. That's a 1D string, right? You can also look at it in terms of secondary structure assignment. You can say that at a particular position there's a helix, there's a helix, there's a helix, and that simply is a string again, 1D, okay? You can look at a feature that is called solvent accessibility, where you simply ask, this is my protein, it's a sphere, and some of these molecules in the protein are outside, some of the molecules in the protein are inside, and you're simply going to have a measure percentage of the protein of this particular amino acid to be inside or outside, okay? And that's, again, is a single number for every single position, and therefore it's a 1D piece of information, okay? Then there's a 2D information. The 2D information is essentially looking at every single residue in that protein. In this particular case, the protein here shown has 98 residues. You show protein residue number 1, or amino acid 1, 2, 3, 4, 98. Here again, and it's black. If these two amino acids in 3D shape are close to each other. So on the diagonal is everything is black, because protein I and I plus 1 are close to each other in 3D. Simply because they follow each other. And then there are sort of things that are far away. So here are contacts between residue 80 and residue 20. Okay? So there are things that are far away in 3D structure, far away in sequence, but close in 3D structure. Now, this 2D information, that's what I said, you can compare 2D maps with each other. But this 2D information is one way of looking at the protein information, right? This is 1D information, 2D information, and there's the 3D structure here. The 3D is simply the 3D coordinates. Okay, now in terms of simple dimensionality, the moment I go from 3D to 2D, I lose information, right? This is supposed to be a question. I want an answer. There are two possible answers. One is yes and one is no. And they both may come, whoever answers that is supposed to sort of speculate why.
Yes.
Yeah?
Yes, because,
Sorry. Yeah?
yes, because you lose a dimension when you go from
You can reduce the dimensions, but do you lose information?
you lose information,
You lose-
you lose on a dimension. When you
You lose a dimension, and therefore you lose information.
Yeah.
Same thing. Now, let's think about this. So, before I looked at the sphere that had 8 points, and I represented the sphere with 8 points in a matrix. And I said in every single entry in this matrix you show how close all the other 7 points are. And I'm doing the same thing here. What is it that you do not capture of the 3D information in this image? And the answer to that question actually is only the image and its mirror. So you cannot distinguish between this and the mirror image of it. That's the only piece of information you lose, which is not a dimension. Essentially, if you have the complete distance matrix, so the complete 2D info, that contains almost, or essentially as much information as the 3D. That may be very counterintuitive, but if you think about it in terms of shapes, of simple shapes, you will see that although you go from 3D to 2D, you lose a dimension, but you're actually not losing the information. Think about it on your way home about simple images. It's actually very simple, very simplistic, and you sort of can think this through very easily. When you go from 2D to 1D, that is when you lose a lot. So in the 1D string of secondary structure, for instance, you know that a particular position here, in this particular case they happen to be only beta strands, they're labeled with an E. So you know where there are these arrows, but you do not know which arrow connects to which arrow. So you completely lose the 2D information. And with losing the 2D information, you can never from 1D get back to 3D. From 2D you can. If you have a complete 2D map, you can get. Is that a question that others would care about? Most likely the answer will be yes. So can you ask it, please?
I didn't get completely why we don't lose information.
So let me repeat this. No, not repeat this. Let me make this more clear. I did not prove to you why you do not lose information when you go from 2D or from 3D to 2D. I simply stated, here I am standing, I'm the big professor, believe me. There's no way for you to understand it. I believe that if you go home and think it through, not with a protein in your head, but with a very, very simple cube or something, you will be able to find out that in fact in the 2D matrix of a cube, and you guys, some of you have done vector graphics, this is where you use this. You put essentially an image, a 3D image, into a 2D vector. And this 2D vector is not an approximation. It's the real 3D image. The full information about the 3D information is in the complete distance map, yeah?
You said that the 3D structure all the distance between each
Yes. So when you take the 3D structure, again, the 3D structure in this particular case is a protein with 98 amino acids. By the way, I keep talking about residues in amino acids. So there are 20 different amino acids, and when you put them together in a protein, we tend to refer to them as residues. So there are 98 different residues in this particular protein. And where every single one of these is in 3D, that's the 3D structure. The 2D information is simply you take every one of these 98 and you ask all the other 97, how close are they to this one? Okay? So you take the first and ask all the 97, what are they close to? And so forth and so on. You do that at every single point. The matrix here obviously is symmetric. So I being close to J means that J is close to I. That's why it's symmetric. But then, so essentially you only need the upper diagonal. And again, the only thing to reproduce from the upper diagonal, you can completely reproduce the 3D info except for its mirror image. And again, I did not prove that. On this one, you have to believe me. And again, I believe you can, yeah?
Is not being able to tell the mirror image not important for proteins?
So the mirror, so distinguishing between the protein and its mirror image, typically we would argue, so for most proteins, we would recognize which of the two you have in front of you, because only one tends to be the major thing in nature. And that has to do, ultimately, I said, that is N to C, that has to ultimately do with the chemistry of the way they are strung up. There are exceptions to that, and this statement is not as true as we have, over the last 10 years, found several exceptions to that. But overall, we still believe that this is sort of a probability, in a probability sense, is likely true, this statement. Okay, so then you can, if the 2D contains the same information as the 3D, you can do 3D alignments by doing 2D alignments. And there's one particular method here called DALI that does it. And the trick in DALI is exactly, the problem in DALI is exactly the problem that we talked about before, which is the problem, when do I stop cutting? So I said it's very easy with the 2D to compare 2D matrices because you can compare subsections of these matrices. But the question is, how small do you get? Do you really get down to the level of the amino acid? You know that you're not going to do that. But where are you going to stop? I said you can look for scientific significance, statistical significance. You have a background distribution, but it's not that easy. And that's the problem with this method. There's another idea here that comes from Munich, from Fabian Brützel and Rolf Zimmer, called Voronoi, because essentially it creates Voronoi environments. and describes essentially every, so here 1, 2, 3, 4, 5, 6, 7 residues here. It describes these 7 mirror and these 7 mirror, these 7 residues by, it looks at the X and compares the X and Y according to its environment that is indicated by the Voronoi environment, and then essentially it aligns Voronoi environments. The issue that I talked about before, the problem of global, yes?
You're using dynamic programming, so why don't you use a stochastic algorithm?
Why don't you use dynamic programming for cutting? Is that what you're asking?
Yeah.
So, why do you talk about dynamic programming? Does everybody in the room know dynamic programming? Oh! Here I am. So before this, there was a slide on dynamic programming. One of the first structural alignment methods used dynamic programming. And I cut it out because I felt it was too complicated, the slide. I didn't remember that. So what is your question?
That you always try to compute the exact solution, not the approximation.
So so when you cut it into Voronoi environments and then you do an all against all comparison, then you need a dynamic programming simply because of the complexity of the numbers. So you may call that a hack, if you want. So you have too many possibilities. Is that a good? But we're getting into the dynamic programming. Then it's a good solution, but it's not. So you have to still prove that the dynamic programming in fact maps. So for this particular case here, the proof is not given. So in this particular case, it's a hack. I'm going to talk about sequence comparisons, and for those under certain assumptions, the dynamic programming will find the optimal solution. But dynamic programming is a shortcut, right? And you need a shortcut because it's combinatorially explosive. Is that answering your question? No, it's not. Then I didn't understand the question. We have a one-on-one. Yes?
Does one protein always fold in a similar way?
So the question is, does the same protein always fold in the same way? And ultimately the answer, again, is a simplification of the real answer, but in principle the answer is yes, if not disease. And in fact, a lot of our proteins are occupied with trying to interfere when proteins fold incorrectly, because that leads to disease. Prion, the mad cow disease is one example where proteins essentially don't fold the right way. And the sort of immune system is not able to correspond or respond to that. It's not an immune system. It's the inside of a cell. There are so-called heat shock proteins, and you can think about those like an immune system. It's essentially proteins that go out and sort of go around proteins that are not correctly folded. Help them to refold or take them apart. It's an internal control system. So the issue really is one between what there are two forms of calmodulin here. One is ligand free, one so bound to something, one is bound not bound to something, bound to something in blue here. And you see that the same molecule, and that brings us back to the same question before, this and that, the same protein is not folding the same way, you may argue. But that is not true. So without a ligand, it will always look like this. With a ligand, it will always look like this. So the same protein will always fold the same way and will always bind the same way. Okay? And this again, I said, if not then disease, this is again very important because shape is function. If you don't adopt the right shape, you cannot do the right function. And so capturing this difference is in fact something that is not easy to do, and it's not easy to do it if you compare three-dimensional objects. You have to cut them, and in cutting them you have to see what is the part, what are the parts, what are the local regions that superpose that in fact are corresponding. There are many, many, many other methods that align proteins in 3D. I just want to mention that. Short recap. Cells are surrounded by membranes. These membranes let stuff in and out. Inside the cell is extremely densely packed with, for instance, proteins, mostly proteins. Proteins are the machinery of life. They come in different shapes, different sizes. Some of them are tracks. Some of them proteins, in this particular case here, are on the surface. There's an HRV and a human T cell. So that is in fact the immune system. And in this particular case, the way, in this slide from Natasha Wood, who is doing simulations here on these molecules that stick outside of the cell, when the HRV envelope protein, so the envelope protein is the one that is around the HRV virus, attacks. And when these two molecules attack, it turns out that one important part, in order to understand how this interaction is done, is a region that is called a loop. So it's neither helix, the rods, nor beta strand, these arrows, but it's something that is flexible and movable. And these are regions that typically are considered in the world of proteins in terms of structure. The helices and the strands, these arrows and the rods are important, most relevant to describe a protein structure. What is people most focused on that, and typically they ignore those regions that somehow stick out. In this particular case, a few of the regions that stick out are essential for the interaction, essential to understand how HIV enters the body. So-called V3 loop, and in fact her research is trying to model this V3 loop and trying to understand how HIV attacks. There are 3D modules that build up one way. They build up proteins. One way of thinking about it is domains, I said. But in fact, there are units that are smaller than domains. And we can try to sort of identify similarities in 3D between these subunits, these regions.